Hi everyone. This is the third lecture for your chapter Photosynthesis. In the last class, we discussed what all different kinds of pigments are involved in photosynthesis. Okay, we discussed the carotenoids as well as xanthophylls. Then we saw chlorophyll A, chlorophyll B. There are two or three more varieties C, D, and E, right? Where all they are found, in which all species, what kind of chlorophyll is found. We also saw some basic information about this whole process of photosynthesis, right? We saw, let us just start with today's lecture. A little bit of revision. We saw that photosynthesis, it takes place in half of it in the membranous system, which includes the grana, thylakoid, stromal lamella, and the other half of photosynthesis happens in the stroma, okay, the matrix portion of your chloroplast. We also had seen the structure of chlorophyll, chloroplast, of leaf, right? So in today's lecture, we will be seeing mostly about the light reactions. Let us see what these light reactions are. Before that, what part of photosynthesis takes place in the membranous system? So this membranous system is responsible for trapping light. Okay, it is the portion of the chloroplast which will trap light for synthesizing ATP and NADH. Okay, NADPH, sorry. So ATP and NADPH can only be formed if you can trap some light, convert it into chemical energy, right? So this happens by with help of chlorophyll. Okay, that also we had seen, and chlorophyll is found where? It is found on the thylakoid membrane. Clear? So this phase is directly dependent on light, and hence it is called light phase or photochemical phase. Okay, there is some photochemical reactions taking place. Photo means light and chemical you already know. So photochemical reactions take place here. That is why it is called light phase or light reaction, as well as photochemical reaction. Okay, they take place inside the thylakoid, in the grana, or in the stromal lamella. The second part of the photosynthesis takes place in the stroma. It is all the enzymatic reactions which are reason for synthesis of sugar. Okay, so these enzymatic reactions are nothing but your Calvin cycle, which we shall study in coming lectures. So Calvin cycle is basically made up of multiple enzymes which convert carbon dioxide into sugar. It is not directly dependent on light. As you can see, there is no direct light which is required for this phase. Okay, only the products of the light reaction, which are NADPH and ATP, are required for this phase. Okay, so it is also known as dark reaction or Calvin cycle. Okay, starting with the light reaction or Hill reaction, or which is also known as photochemical reaction. We had seen these different functions, okay, these different parts of light reaction in the last class. This is where we ended. So in light reaction, you have these four things happening. What are the four things? First thing will be the light absorption. So there is light which is being absorbed. Second thing, there is water splitting. So H2O molecule, it is getting split into H plus and O2. Okay, so this O2 which is formed will be released. So there is oxygen release, and finally there is formation of high energy intermediates. Okay, these excuse me, these high energy intermediates are nothing but ATP and NADPH. Okay, fine. NADPH, I hope you already know, but still once more, it is nicotinamide adenine dinucleotide phosphate. Okay, the whole molecule is known as nicotinamide adenine dinucleotide phosphate. Fine. Okay, moving ahead with the light reaction, let us see it in detail. In light reaction, you have the pigments, okay, the photopigments which we saw in the last class, which included the chlorophyll, xanthophylls, carotenoids, all of those. They are organized into a photochemical light harvesting complex, okay, which are called LHC. So these pigments, they are not just randomly arranged on the membrane. Fine, they are organized in such complexes which are called light harvesting complex. The light harvesting complex are made up of such pigment molecules as well as the protein. So these purple colored structure that you are seeing, okay, this is a cross section. You can see multiple such purple colored structures. Each of these are light harvesting complexes. These green dots that you see are nothing but different photopigments. Okay, it could be chlorophyll, it could be xanthophyll, it could be carotenoid, anything. And the rest of the purple portion is made up of protein. So in light harvesting complex, there are hundreds of pigment molecules. Okay, there can be hundreds of pigment molecules which are bound to the protein. So this here one whole thing is called one light harvesting complex. In this diagram, you can see three light harvesting complexes, right? 1, 2, and 3. Is this much clear? Okay. Each light harvesting complex has all pigments except chlorophyll A. So remember chlorophyll A, we will call it the reaction center chlorophyll. Fine. So apart from chlorophyll A, what all pigments remain? So you are left with xanthophyll, you are left with carotenoids and chlorophyll B. All of these are present in the light harvesting complex. Apart from this light harvesting complexes, which are the purple colored ones, you are also able to see this light blue colored complex. Can you see? This light blue colored complex is nothing but your reaction center complex. What is reaction center complex made up of? It is made up of a pair of chlorophyll A. So there are two molecules of chlorophyll A. Clear? This chlorophyll A will be present nowhere else. It is only found in the reaction center. So here is a chlorophyll A, and the chlorophyll A, it will transfer its electron to some acceptor. So that primary electron acceptor plus molecules of chlorophyll form the reaction center complex. Clear? The whole complex together, the whole system with light harvesting complex plus the reaction center is known as photosystem. Okay, many students have this doubt. They tend to get confused between the difference. Okay, what is light harvesting complex and what is photosystem? So here is a clear explanation. As I told you, protein bound pigments, okay, except chlorophyll A, the total pigments, they are known as light harvesting complexes. In the center you have two chlorophyll molecules together and they transfer electron to some electron acceptor. This whole complex is known as reaction center complex. Clear? So two parts, light harvesting complex, reaction center complex, and together, these both together, they form photosystem. Clear? Fine. As you can see, this is your thylakoid membrane. So we are still discussing where. So that where is on your thylakoid membrane. Clear? As I had already told you, the pigment molecules are present on the thylakoid membrane. And this is how they are present. Clear till here? Very good. One more thing I wanted to tell you. The other pigments, okay, except chlorophyll A. Remember, they are also known as accessory pigments or they are called antenna. Clear? They are called accessory pigments or antenna. Fine. Let us start with the next part, which is photosystem. It is light harvesting complex which has pigments plus proteins plus the reaction center which has chlorophyll A. Clear? So this photosystem, it includes all of these parts. Fine. Chalo. Now this photosystem can have two types. There are two types of photosystem. Photosystem 1, which is in short written as PS1, and you can have photosystem 2, which is in short written as PS2. How they were named? So they were named in the line of discovery. Okay, PS1 was discovered first, PS2 was discovered later. So it was given the name 1 and 2. PS1 has chlorophyll A, which absorbs light at 700 nanometer. Remember this? Also in the last class we had seen the absorption graphs, okay, the absorption curve. So in chlorophyll A also there are two varieties. Fine. In chlorophyll we saw there can be many. Chlorophyll A, B, C, D, E. Now in chlorophyll A there can be two types. There can either be chlorophyll A 700 or chlorophyll chlorophyll A 680. Okay, so chlorophyll A also has two types. The chlorophyll 700 or chlorophyll A1 is the type which absorbs light at 700 nanometer. Okay, so all the 700 nanometer light will be absorbed by chlorophyll A of photosystem 1. In case of photosystem 2, the chlorophyll A absorbs maximum. Okay, so it has absorbance peak at 680 nanometer. So it will be absorbing more light in the 680 nanometer wavelength. Clear? How I remember this one and which is 700, which is 680? Just remember the bigger number. Okay, 700 is bigger than 680. So bigger number is given number 1. Clear? You will always give number 1 to the student who scores the best, right? So 700 is a larger number, is a greater number. So you give it number 1. So photosystem 1 has 700 nanometer um, light absorbing chlorophyll A, and PS2 it comes second. Okay, so 2 you can also use to remember that there are 20 nanometer less compared to the first one. So 700 minus 20 will give you 680, and if you can remember 680 just like that, well and good. So PS2 is 680 nanometer. Clear? So two types of photo system. Right, so this is how it is happening. Okay, this light or the photon is nothing but the particle nature of light. As you may have already studied, light has dual nature, particle as well as wave. So here the photon, which is the particle nature of light, is considered. Photon may fall on such chlorophyll B molecules. These are all chlorophyll B molecules, green in color. Then this red one is nothing but your carotene. Okay, it can be some different type of carotene. Then you have these orange colored molecules, which are also a different type of carotenoid. And this yellow one is your xanthophyll. Together they are forming the antenna or the accessory pigment. Have you seen any satellite that they show uh, which can be like this? I am pretty sure you must have seen. You've seen such kind of a satellite, right? And it may have a base like this. Okay, scientists use this as telescope to find out different radiation from the atmosphere, from the space, right? So just think of it like this. You know why this disk is there, this C-shaped disk? It is just there to make sure that all of the radiation that is coming may fall on this main center. Okay, are you able to understand? This is your satellite. Okay, and that satellite will have some cone shaped thing in between. Fine. And the C-shaped structure is doing what? It is just helping whatever radiation will be falling on it will be reflected and focused on this center. Clear? So it is allowing to absorb more and more information from this small part. Fine. Same thing is happening in case of our photosystems as well. Okay. Here you can see this is acting as an antenna. It is collecting all the photons, it is collecting all the energy, and this whole energy, okay, all the the energy gotten from the photons will be transferred finally to the chlorophyll A. Just here you can see this is the light falling, the photon. You can see there is transfer of energy, and this energy is finally given to the chlorophyll A molecule. Clear? Which is the reaction center molecule, reaction center chlorophyll. And this, when it gets excited, it will transfer its electron to the electron acceptor. Fine. Now, how do you know there are two different photosystems? Like even if I tell you, how will you take it? Okay, how will you just assume it? So the scientists, they actually did an experiment. This is the sort of experimental setup. They took a flask. So here is your flask. It has water, and inside water it has algae, which is called chlorella. Clear? It used chlorella algae, had water, and this is the chlorella solution. Now to this chlorella, they provided it with a flash of light. Okay, for 10 raised to minus 5 seconds, there was some light which was given. After that, there was measurement of oxygen which was released. Okay, Right. Now let us see this oxygen release. Of course, as you may remember from the graphs, it can be used to measure how much amount of photosynthesis is taking place. Clear? Um, so when they gave a 680 nanometer flash, okay, which means the light falling on this chlorella was of 680 nanometer, the amount of or the rate of photosynthesis was this much. Same thing when they dried with 700 nanometer wavelength, the amount of photosynthesis was this much. Okay, this is 680 nanometer, this is 700 nanometer, and as you can see, they may have already tried with different other wavelengths also, but they got peak only at 680 and 700 nanometer. Fine. After that, when they provided with 680 and 700 together, so now this light has 680 nanometer as well as 700 nanometer. They saw that the rate of photosynthesis increased 3 to 4 times. Okay, so this here you can say that 680 and 700, both of them are required for photosynthesis, and this separate peaks, they showed that there are two types of chlorophyll A. Clear? So this explained the presence of two types of photosystem. Clear? Immersion red drop experiment. Right, one more thing. This is sometimes also referred to as red drop because it was done on a drop. Okay, um, I forgot that part. So you can have some sort of such handle. On this handle, they put a drop of water with chlorella. Okay, so it had chlorella and they put a drop of water. And on this drop of water, they gave 680 and 700 nanometer wavelength light. Okay, and then they carried out this experiment. So it is also sometimes referred to as Emerson red drop experiment. Fine. Right. So now, where are these photosystems located? Okay, where can you find these photosystems? There are two types, photosystem 2 and 1. 2 you will mostly find in this folds. Okay, wherever there is such a pressed part. Remember, thylakoid is not thylakoid are these coin-like structure, and then grana is nothing but a stack of such coin-like structure. So in this stack, whatever these portion, okay, which are between two thylakoids, they will be having photosystem 2. Here can you see this? Here is one thylakoid, second thylakoid, third, fourth. So wherever there is some what you can say connection to the next thylakoid, or where it is touching the next thylakoid, you will mostly find photosystem 2 present there. Fine. And wherever it is free, the thylakoid membrane is free, it is not touching any other thylakoid membrane, you will find photosystem 1. Okay, these are all photosystem 1. This here is called the pressed part of the grana. Okay, because it is pressed, it is sandwiched between two layers. And here these parts are called a pressed part. Okay, opposite of pressed is oppressed. It literally means not pressed. Clear? So it is the oppressed parts of um, your grana where you find photosystem 1. Clear this much? Knowing all of this, let us now see the difference between photosystem 1 and 2. Photosystem 1 has reaction center chlorophyll A, which gets excited by 700 nanometer red light. Photosystem 2 has a chlorophyll A which gets excited by 680 nanometer of red light. It was discovered first, hence it is called PS1. Photosystem 2 was discovered second. Photosystem 1, it is present in non-oppressed part. Okay, it is present in the free part of grana, while the photosystem 2 is present in pressed part of grana. From this you can even say that in stromal lamella you will find more of photosystem 1. Clear? Fine. Photosystem 1, it participates in cyclic as well as non-cyclic photophosphorylation. If you do not remember this for now, it's fine. We will be discussing this later. So just remember this point, okay? That it participates in cyclic and non-cyclic photophosphorylation, while photosystem 2 It can only participate in non-cyclic type of photophosphorylation. The photosystem 1 does not take part in photolysis of water. Okay, it will not break down the molecule of water, while photosystem 2 is the only system which will break down the water molecule. Clear this much? So these are the differences between the two photosystems. I hope till now you are clear with what are light harvesting complexes, what are photosystems, and what are reaction centers or reaction center chlorophyll. Fine. Then let us move on to the first part of light reaction, which is photolysis of water. Okay, we have seen there are two major parts. One is photolysis of water and evolution of oxygen, and second part is conversion of the light energy into some high energy intermediates. Fine. So beginning with photolysis of water, it occurs inside lumen of grana or lumen of thylakoid. Okay, so if this is the thylakoid, this photolysis will take place inside the lumen, okay, inside this hollow cavity of the thylakoid. Clear? Remember these points, okay, they are necessary and we will come back to those later on. So they occur inside the lumen of thylakoid or lumen of grana. Only PS2 can split water, only photosystem 2 can do this function of splitting water. Here, three ions are very important. Okay, remember these because even when we discuss mineral nutrition, okay, which is another chapter, there also this will be mentioned. Okay, so three mineral ions are very important. One is manganese, Mn. Remember, not magnesium, manganese, Mn. Calcium, Ca2 plus, and chlorine, Cl minus. These three are very important for this reaction. Now just looking at this diagram, let us understand this diagram. Fine. Here you have these P680, P680 are nothing but your photosystem, okay, photosystem 2. Photosystem 2, as I already told you, are the only ones which can split water. Photosystem 2, you can also write it as P680. Fine. Okay, now what happens is, this is present in your thylakoid membrane, outside is stroma, inside is lumen. 4 photons, when they fall on this photosystem, they will excite this photosystem, they will release electron. Okay, so there will be electron deficit in these photosystems. Okay, the chlorophyll A will be lacking electrons. Clear till here? Fine. Now let us see our photolysis part. Now this photosystem has a special group of proteins beneath. Okay, these are different subunits of the protein. Their weights are given: 17 kilodalton, 23 kilodalton, 33 kilodalton. These are the different weight of the proteins. So 3 or more subunits of such proteins, they come together and form oxygen evolving complex. Oxygen evolving complex is the complex which will release oxygen. Clear? In this oxygen evolving complex, there is a subunit which requires these 3 ions. You require manganese, you require chlorine and calcium. So now can you see where they are exactly present? They are present in the oxygen evolving complex, which is sometimes referred to as OEC. Fine? Okay. This is again present near the thylakoid lumen, which is the inner portion of the thylakoid. Fine. Now what exactly happens? Inside the thylakoid lumen also there is water. Okay. So 2 molecules of water, they will go and they will be broken. Okay. They will be lysed. How will they be lysed? This will happen in presence of light. Okay. Only sunlight can help it. That is why it is called photolysis. Okay. Lysis is breakdown. And in presence of light, so you call it photolysis. In presence of light, this water molecule will be broken down into protons, so 4H plus oxygen. Okay, and this oxygen is the one which is released. So this is the first step where you are getting the first product of photosynthesis, which is oxygen. Clear? So this is where the oxygen is released. Looking at the balanced reactions, let us look over here, come back here. You require 4 molecules of water, so 4H2O will give you 4H plus 4OH minus. Again, now this 4OH minus, what they are doing further? Okay, you have still not gotten oxygen directly. What happens now is, 4OH minus will give you, this minus will become 4 electrons. Okay, so you get 4 electrons plus 2 water molecules plus 1 molecule of oxygen. Clear? This OH minus, 4OH minus, they will come together. They will give you 4 electrons, 2 H2O and 1 oxygen molecule. When you balance this, okay, when you cancel it out, the net reaction of these two will be, as you can see, 4H2O here and 2H2O here. So you have on the left hand side 2H2O. Clear? You can cancel these. I hope you know how to balance these reactions, the net reactions. So 4H2O here and 2 here. So 2 will be cancelled. Here you get 2. That is what you get here. 2H2O is giving you, now there are 4 OH here and 4 OH here. So they are directly cancelled. Fine. So on the left hand side you are left with nothing. You just have 2H2O. 2H2O is giving you 4 electrons plus 4 protons plus 1 oxygen molecule, which is released, okay, which is why this upper arrow. Fine. So this is how the photolysis of water is taking place. Photolysis of water, only PS2 photosystem 2 can do it. Photosystem 2 also has this special oxygen evolving complex. In oxygen evolving complex, 3 ions are important: manganese, chlorine and calcium. In presence of these 3 ions, only the net reaction is written, which is 2H2O will give you 4H plus plus 2O2. And where are the electrons, you ask? These here are the electrons, okay? The 4E minus, the 4 electrons are shown here. Clear? Fine. Next time we will be talking about the second part of your light reaction, which is the electron transport chain and photophosphorylation. Okay, see you next time.